Have you ever wondered how we are able to see the beauty of the land around us, the landscapes and the people? A simple answer would be our eyes. But how do our eyes see? Behind the lens of our eyes, there is a thin sheet of different types of cells. This is the retina. The retina contains rod cells. These cells consist of synaptic endings, nucleus, inner body, outer fragment, pigment pockets. Rhodopsin is a special pigment present in these pigment pockets. Rhodopsin contains 11-cis-retinol, which is converted to all-trans retinol when light falls on it. The rhodopsin in the cell membrane is present in association with a protein known as transducin. When light falls on rhodopsin, the 11-cis-retinol is converted to all-trans retinol, leading to the change in conformation of the rhodopsin. This activated the transducin and allows it to act in closer proximity to the rhodopsin. The GDP that is bound to it is converted to GTP. This GTP binding causes the alpha subunit to lose its affinity for the beta and gamma subunits, leading to the removal of alpha subunit from the receptor as the gamma subunit help to dissociate with the receptor. The beta and gamma subunits diffuse away while the alpha subunit approaches an enzyme called as cyclic GMP phosphodiesterase. The binding of G protein subunit causes the inhibitory subunit of phosphodiesterase to be removed, causing its activation. cGMP phosphodiesterase enzyme converts a molecule of cyclase GMP to GMP, increasing its concentration in the cell. Outside the rhodopsin pockets, cGMP-dependent transporters are present that allow the influx of sodium ions into the cell. This maintains a depolarized state in the cell. In the presence of light, when the cGMP is converted to GMP, the cGMP-dependent sodium transporters are inactivated. This causes hyperpolarization inside the cell due to constant efflux of potassium ions. In this hyperpolarized state, the amount of neurotransmitter is less. This neurotransmitter is glutamate. This decreased release of the inhibitory glutamate neurotransmitter results in the bipolar neurons to release their stimulatory neurotransmitters to activate the optic nerve. This causes activation of the occipital lobe of the brain, resulting in vision. When the calcium concentration decreases in the cell, guanylyl cyclase is activated, which converts GTP to cyclic GMP, inactivating phosphodiesterase. As cyclic GMP concentration increases, sodium and calcium transporters open, letting the influx of the atoms and allowing the cell to return to its initial inactive state. The GTP of G protein is hydrolyzed due to intrinsic GTPase activity. The G protein is removed. The inhibitory subunit reattaches to the phosphodiesterase. The all-trans retinol is converted to 11-cis retinol, making the rhodopsin available for another cycle.